Good afternoon and welcome to Bulldogs Behind the Scenes. This series gives us a closer look at the people, places, and research connected to the University of Minnesota Duluth. Today we are diving into a specific topic that affects us all, plastics in our environment, and learning how they break down and persist over time. Before we begin, let's take a moment here to let everyone finish logging on. Well, my name is Molly Clevin, and I'm with the UMD Alumni Relations team, and I'm so glad you could join us. Today, we'll be going behind the scenes with Dr. Melissa Maurer-Jones to learn more about her team's research on microplastics and how these materials behave in the environment. Before we get started, I want to mention a few of the other places we've explored through the series: the Minnesota Landscape Arboretum, Amsoil Arena, the Superior Hiking Trail, the proposed UMD Sales Center, the UMD Chocolate Lab, Romano Gym, and more. If you missed any of these, you can catch the recordings anytime at d.umn.edu/alumni. Today's session is being recorded and will also be available on our website. You'll get an email with a link once it's ready. We'll have time for questions at the end, so as you're listening, feel free to drop your questions in the Q&A box at the bottom of your screen. Now I'm excited to introduce Dr. Melissa Maurer-Jones. Melissa earned her Bachelor of Science in Chemistry from the University of St. Thomas and her PhD from the University of Minnesota Twin Cities, where she studied nanoparticle toxicity. She went on to complete her postdoctoral research at ETH Zurich, focusing on the environmental photochemistry of plastics. Since 2016, she's been an associate professor in UMD's Department of Chemistry and Biochemistry and currently serves as Director of Graduate Studies for the Chemistry Master of Science program. Melissa, thank you for being here, and I'll hand it over to you.
Thank you so much for the introduction, Molly. And I'm really excited to be here today to tell you about some of the work that we're doing here, both specifically in my lab, but then generally at the University of Minnesota Duluth, looking at microplastics in the environment and generally the aging of plastics. I know Molly started out by telling us a little bit about my journey, but I like to kind of start here because I think it helps kind of center what I think is how I view this problem or these research questions and what I'm interested in studying in my lab and beyond. So I started my kind of academic journey doing a bachelor's in chemistry from St. Thomas in St. Paul, Minnesota. I ended up taking a gap year because I wasn't quite sure, even though did research in an analytical chemistry lab for 3 years, 4 years in my undergraduate. I wasn't quite sure what I wanted to do next. And I thought a little bit about potentially going a policy route, and I did some time with a nonprofit called Environment Georgia, just trying to kind of understand what that meant and really realize that as a scientist, that I can make a big impact on our understanding of humans' impact on the environment by doing good science. And so that led me back to wanting to ultimately get a graduate degree. I took a little time, as you know, the timing of everything on an academic calendar doesn't always match. And so I ended up taking a little time working in industry. I worked for Pace Analytical, but was placed at 3M and had an opportunity just to learn what it meant to do some work as an analytical scientist in an industrial setting. I went on to do my PhD at the University of Minnesota Twin Cities, and I worked with Dr. Christy Haynes. And the focus of my research was really on the toxicity of engineered nanoparticles. So in this work, I kind of combined areas of material science, specifically engineered nanoparticle synthesis and characterization. I learned how to do some toxicity testing on both a mammalian cell and an environmental ecological bacterial model. But most of my work was centered around applying sensitive analytical technology to understand the toxicity response of these biological systems to these engineered materials. So these themes of material science, analytical chemistry, some biological work, I carried that on into my postdoctoral research at ETH Zurich. where I worked with Dr. Chris McNeil, looking at another material, polymeric materials, and we were trying to understand the breakdown of a biodegradable plastic under relevant environmental aging conditions. And specifically, I was looking at what if sunlight shone on these plastics? How would that photochemical breakdown change its biodegradability? Um, that work, again, that work was highly interdisciplinary. I was focused on material science again, but in this case, it was polymers. I was doing some aquatic photochemistry and environmental chemistry. Again, all with this lens of thinking about analytical chemistry and doing good measurements. And so I was fortunate to land a tenure track faculty position here and I started in spring of 2016. So I'm just coming up on my 10-year anniversary at being at UMD and really excited to talk about all of the work that my group has done so far and hopefully give you a glimpse of what we're thinking about moving forward. Because similar to my PhD and my postdoc, I'm bringing themes of material science, of environmental analytical chemistry, photochemistry, all with this lens of analytical chemistry into my current research that I'm doing right now. So let's jump into kind of the topic at hand. And I think a lot of people here decided to join because it is really a hot topic in sort of the public awareness. So just to get us started, I want to make sure we have some common language because, you know, as PhDs, we sometimes slip into very specific language that we use. And so I want to make sure everybody is kind of following along. So the first thing I like to emphasize is that plastic is a mixture. Its primary molecule is a molecule called a polymer. And this is usually is a hydrocarbon, a carbon and hydrogen molecule. And the different hydrocarbons, these polymers are what give us our recycling codes that are what we often look at to see if our recycling facility will handle it. So all of the materials that have, say, a recycle code 1 are all made out of the same polymer, polyethylene terephthalate. And sometimes, and so while the polymer might be the same, it might all be number 1, we know that sometimes we get bottles that have a number 1 on it that are different colors or have different like kind of flexibilities to that. That speaks towards the fact that plastics are a mixture. It's not just that polymer molecule that's in this product. It's also a group of additives that are in there as well that are used to adjust or adapt the material properties. So it might be pigments, or it might be something like antioxidants or plasticizers, all of which will change kind of the feel of the material, the look of the material, the performance of the material. So microplastics, and even something called nanoplastics has been something that people have been talking about in the news, I would say pretty robustly for the past 3 to 5 years. But the definition of what that is a microplastic is a particle or a small particle of plastic. The size range of what a microplastic is something that's between 1 micrometer and 5 millimeters. That's just kind of what the field has defined as a microplastic. A nanoplastic is something that is smaller. It's a particle that is less than 1 micrometer in its longest dimension. And so why are we talking about micro and nanoplastics? Well, where do these come from specifically? The primary route or the main way that micro and nanoplastics end up in our environmental systems and in us, and they exist because they primarily come from the breakdown of larger plastic materials. Okay, so let's look at this little flow chart here. So this is kind of to represent that we live in a world that uses a lot of different plastic materials. We have different industries that maybe are manufacturing plastic that might have spills here. There are some personal hygiene products that have plastic little pellets in there as exfoliants. We also wear synthetic textile clothes. We use these other materials that break down, and ultimately that ends up in the presence of micro and even nanoplastics into our environment. And from here, these microplastics, can go. They can be taken up by fish. They can go down and be in our sediment. They could be these fish could be caught for us to eat, or they could be, consumed by other wildlife. And ultimately, these micro-sized plastics can have this movement around our environment that, that may have an impact. This is an example of a nutrient cycling, or specifically a carbon cycling. And in the last, I would say, 5 years, there's been a big push to understand how much does plastic contribute to the global carbon cycle. So the way that we look at these, as a chemist, I trained as a chemist, and I didn't really know how to look at these until I met some biogeochemists. So just for those folks that don't know how to look at this kind of carbon cycle flow chart here, if it's a rectangle in white that and in white just text here, these are reservoirs where carbon can be sequestered, right? So it can be in our plant biomass, or it can be in our sediments here. The arrows and the yellow text is relating to the flux of carbon. And one thing we're becoming more aware of is that plastics are a large source of carbon, and they don't seem to be transforming and moving to different reservoirs very quickly necessarily. And so we end up having an kind of these plastics in all of these different reservoirs, and we're not quite sure how much is there and how much is plastic contributing. You see a lot of arrows in this flow chart. We also don't really know like how fast they're moving and fluxing through the environment. And so, and microplastics and nanoplastics are probably the form in which these plastics are really moving through these different systems. And why do we care? Why do we care about plastics environment? Why would this be a problem that plastic is there? Well, I grouped this into three different kind of risk categories that we're really starting to unravel why the plastic being there might be problematic. There's physical risk. I think that we've all seen photos or videos of wildlife or macrofauna being caught up in plastic netting. I think that, I grew up in the 80s and early 90s, and the big campaign to cut your pop container, your pop plastic rings up so that we don't trap turtles. That's definitely part of it. There's a physical risk also in that these plastic materials, including microplastics, can be consumed by by different organisms. And if they're large enough, what they can do is they can't necessarily pass through the digestive system, and they cause something called false satiation. So it makes the organism feel full, even though it really hasn't eaten enough nutrients to be able to sustain itself. So physical risk. There's chemical risk associated with this. And there's a couple ways that I envision that chemical risk happens with plastics in the environment. We have, as I said, plastics are a mixture, and it has additives incorporated into them. And these additives are not necessarily like covalently bound to the polymer molecule. And so what this means is that these additives could ultimately leach out. And we've kind of thought about this, I think, as a community for a while with bisphenol A. Including the Bisphenol A Reduction Act that like removed BPA from a lot of particularly infant bottles and things like that. But there are other additives that may be of concern for the health of humans and other organisms that could leach out from here. The other chemical risk is that the plastics in themselves are quite hydrophobic. They're a hydrocarbon, and they can absorb other organic pollutants onto them or even heavy metal pollutants onto them. And if we think about these particles, these small microplastic or nanoplastic particles floating around, they can then be kind of a transfer, a vector for these other pollutants that are in our environment. The other risk that I categorize is a biological risk. So just like pollutants can absorb onto the surface of these plastics, so could pathogens. And so could other kind of maybe invasive type species. We've already kind of observed this. You think about like zebra mussels and other kind of invasive species that we see in our environments that cause because there's an attachment of these organisms to something that moves around between different water bodies, for example. The same thing can be true for bacteria, for viruses, for fungus. It can be a source of pathogens to be able to attach to and potentially cause a biological risk. And so that biological risk could be if then the microplastic was consumed, or it could be even just moving a pathogen from one location to another. So we should care about answering these questions of where plastics are, how much is there, how they're changing, and whatnot to address these risks that are there. There are lots of analytical challenges, however, to studying plastics in the environment. The first thing I always like to say is that that we're often looking for these plastics in complex matrices. And that's complicated by the fact that plastics are primarily a hydrocarbon, and lots of naturally occurring particles are also hydrocarbons. So to give you an example, we have natural organic matter or particulate organic matter that might be coming from plants or from other organisms that generally happens and into our natural systems. Humus and things like that. That is a natural organic matter. That is a hydrocarbon, just like our plastic is. So we're often looking for a particle that's mixed in with a bunch of other particles that might have a similar chemical signature to the plastics we're looking for. The other issue is that plastics themselves are a complex mixture. And so they often have ill-defined impurities like a residual catalyst at unknown concentration, or they're just generally heterogeneous, meaning that there is microenvironments that occur that they could be even simply from a manufacturing process that they're just a complex kind of material. And that then is going to influence my little picture down here with my pseudo chemical reaction scheme is that's going to influence how these plastics kind of degrade chemically within these systems. So fortunately, I am an analytical scientist and analytical chemist. And what I see as these challenges is a place where really we can dig in and learn and do a lot of cool, interesting things. And so I'm going to move into talking a little bit about the work that I've been doing with my collaborator here, Dr. Liz Miner, that was funded by the Minnesota Sea Grant. And so I wanted to first talk about this work, which is some of the work looking for microplastics and the dynamics of microplastics in Lake Superior, because it really shows that UMD specifically, not just my lab, have become kind of some of the experts in the state for tackling questions about microplastics and plastics in our environment. And so because of these analytical challenges, some of the first questions that we tackled in the last 5 years were, what methods can we use to collect samples at various size ranges and in different areas of Lake Superior and the St. Louis estuary? How do we characterize these plastics? Especially when these plastics are at different size regimes, that becomes an analytical challenge. And how do we ensure that our particle counting is accurate? How do we pull out this information from how do we pull out that we're counting a plastic when we have this complex natural matrices happening? And so these little pictures down here are from graphics that came from a couple of the publications out of this shared project. Some of the things that we worked on was using in situ pumping off of the end of the RV Blue Heron, which is the vessel that's owned by the University of Minnesota Duluth. One thing I like to say is that studying plastics in the environment as a chemist has completely pushed me out of my comfort zone. I had never done field work until I started collaborating with folks here, and they really taught me how do we, as scientists, and who really love our indoor bench science, how do we make sure that it's applicable to a natural system? So I've gotten to spend some time on the boat, and we looked at here using this pump. So what methods can we use to collect? We looked at different pumping regimes. We've looked at different depths of where the microplastics are happening within the water column. And we've looked at comparisons of what is the composition, what are we seeing about the microplastics at different sizes and in different characteristics if we look kind of in the open lake here versus if we look in the kind of Twin Ports Harbor area down here. And so just to kind of show you a little bit of what this field work looks like, there's a couple of different ways in which we tackle this analysis. So we use a lot of efforts of collecting our particles onto sieves, like you saw them pushing the particles off the sieves. But this is one of the main tools we've been using lately to be able to look at microplastic abundance at different depths. And this is a McLean in situ pump. And what you do is we can put filters of different sizes here that we're collecting up, and then we can put it in, store it for when we're ready to do analysis. Microplastic analysis to get away the extra different types of particles and whatnot on there requires quite a bit of sample prep. We go through a bunch of chemical digestion process to basically get away all of the stuff that's complicated about environmental samples, like the natural organic hydrocarbons, and get that away from our sample. So we are left with mostly just plastics remaining. And then we can use equipment, specialized equipment that's shown in this video. This is called an FTIR microscope. And so what this does, an FTIR spectrometer, what it does is it is a tool used to take a fingerprint of the different material that it's taking a spectra of. And that gives us an indication, a fingerprint of what molecules that is. And we can use that to determine if after all of our chemical processing, can we tell that it's a plastic particle or is it some organic matter? Is it a mineral? Is it something, not important? So we can use this high sensitive analytical tool then to kind of raster scan along our sample and kind of look for the polymer particles on there. So that just gives you a flavor of the kind of analysis that we do to be able to look for these microscopic particles with some chemical selectivity in our natural systems. I could spend, multiple papers worth of data telling you all of the different things that we're learning about microplastics in Lake Superior. And happy to talk about that more in the question and answers section. But the reality is Minnesota has a lot of water, not just Lake Superior. And the issue with trying to understand what the impact of humans are in terms of plastic pollution in our waters across the state is really challenging because, like I said, we have a lot of water and all the water around our state is used in different ways. And so we really need to figure out a way to address doing the sampling on a wider scale issue if microplastics ultimately get to the stage of a monitoring effort. So this is definitely something that folks are thinking of, like folks in water quality management are thinking about, is how do we regularly screen for microplastics if this is a emerging contaminative concern that we can ultimately find in our systems. And so my group specifically has been working on a way to produce simplified methods for microplastic collection and detection. And we've been doing it by developing a citizen science sampling kit. that we've been deploying through high school science classrooms. We've also, parallel to this, have been trying to develop some new technology to be able to detect microplastics out of the complicated matrix without large-scale chemical processing. And these two pieces are really important because we need to train a larger network of folks on a simplified method so we can better understand the trends of what's happening around microplastics in our natural systems. And we also need to have faster, simpler, more inexpensive tools to be able to say, yes, that's plastic, or no, that is not. And so where we're at right now on this citizen science effort, we've developed a kit and we've alpha and beta tested these through different high school classrooms, science classrooms. And right now what we're doing is my graduate student here, Abimbola Ige, and has been doing a validation of what the high school students are counting in their sampling and analysis compared to what she counts when she takes it through all of that more complicated chemical analysis with the fancy microscope kind of equipment that we use to detect whether something's a plastic or not. I just put this QR code up here. We have a little website that we have going on to, if any of you want to get this activity into high schools that are in your region, we are interested in partnering with more high schools. So there's more information about the citizen science effort in this QR link. Yeah, you should check it out, and we'd love to connect with other high schools in the state. So far, what I've talked about with you has been thinking about like microplastics as, I mean, generally like something that we're concerned about from the risk standpoint. We're concerned about it because of its prevalence in all of these different types of water, and we're not quite sure all of the sort of health impacts it might mean for us and the environment. But I always like to take a step back because as a chemist, it's really important to understand that we have the power of design. And there is some examples where plastics actually have a potential positive effect on the environment. And so I like to give you some examples. One is agricultural mulch films. Anybody who's a gardener may have experienced this, but on a larger scale too, some produce is produced using agricultural mulch films, which is a mechanical stop to weeds. That's a really helpful thing because we're not applying as many herbicides to our fields. It also minimizes the water that we need because it keeps the moisture in our soil as opposed to letting it evaporate. I really enjoy receiving food and my healthcare supplies as pathogen free. And so that you really need plastic as a way to stop pathogens from growing. And the other thing I think about is even when we think about this carbon cycle, what's the influence of plastics on transportation costs? I did a back of the envelope calculation here to think about what's the difference between if a container, a 40-foot container, had pallets that were either made out of wood or they were made out of plastics. What's the difference in carbon emissions if we had wood pallets in there or plastic pallets? And the answer is substantial. We cut carbon CO2 emissions by a third or a half if we use plastic pallets compared to wood pallets. So what I'd like to say is plastics are actually really an amazing material. There are some indications that it could be helpful in some circumstances about using plastic. The reality is, totally eliminating plastics is probably not something that we are equipped to do anytime in the near future. And so we have to think about how can we use plastics better, and are there different types of plastics that might be a better solution? And so I always like to talk about emerging plastics as being a part of the solution. And when I say emerging plastics, these are plastics that are not, maybe not yet commercially widely used, but are growing in their interest and applicability, and maybe have some disposal mechanisms that are different than simply landfilling or the traditional mechanical recycling route. And so the ones that we primarily think of, and you all might primarily think of, are compostable or biodegradable plastics. in an ideal world, those compostable or biodegradable plastics come from a bio feedstock like corn or something that we can grow. We can isolate our monomers, which are the building blocks for our polymers, and make our resin and ultimately manufacture that into our product that can then go into two different pathways of disposal, including compost that ultimately might be put back onto the field and the cycle can continue, or could go into something like a biogas generation facility, which is something that some municipalities are building, and that we could then use that to produce other forms of energy that we might need. And so these are like an exciting part of the solution for plastics in the environment. That there are still lots of questions about their if they're going to be as kind of persistent in the environment as some of the petroleum-based legacy plastics that have been used for a century. And so having sustainable solutions or sustainable plastics, it requires a complete understanding of the sort of end of life of these products, to make sure that, if they get put in the compost, are they breaking down the way we expect them? Or are they influencing the soil in a negative way? Or can we mechanically recycle these new plastics? And so there's lots of cool questions where my group relies on our analytical materials science approach to be able to ensure that we're using these materials and in a way that they don't become the next generation problem. And so the work that I do in my group, we ask kind of these two types of questions. One of them is how are materials changing chemically or physically under environmental stressors? And then how do these material transformations affect the material behavior? So just to give you an example, one of the things that we've done is we've characterized the chemical reactions of shining sunlight or UV light onto a biodegradable plastic and tried to understand and characterize and quantify the chemical transformation kinetics and the formation of microplastics under these simulated environmental conditions. And then we've gone on to say, okay, we know how these plastics are changing when we shine sunlight on it, which would be a normal thing for a plastic to see. This sees sunlight if it gets accidentally, if you have if a bag gets out into the environment or that agricultural mulch film, it's going to see sunlight. Then the flip side of it is it still going to biodegrade the way we intend it to after we've shown sunlight onto it? Okay. So these are very much so like laboratory studies that we use a wide scope of analytical instrumentations to study. And I just want to give you a flavor for the kind of data and the kind of information that we're working to seek in my lab. And so here's an example. One of the challenges I said like way back on like slide 4 was that it gets really hard to characterize and quantify material transformations in a way that analytical chemists typically do that, because we're really good at doing quantifications in soil or in liquid and in gases, but in solids we don't have as many tools to do quantitative work. And so one of the examples that we have done is we've developed a fluorescence staining strategy to visually see and characterize the degradation of materials based off of the color that the fluorescence glows depending on the degree of degradation. So what we've done is we've used photo degradation or sunlight degradation as kind of our accelerated weathering tool. And then we apply our stain, and if you looked at it with a microscope at the color that these things are fluorescing, at different degrees of degradation, we see different colors because the fluorescent dye we chose to use is solvochromatic. So it's responding to the changing chemistry of the surface of these plastics. And so for folks that want to see some hard-hitting chemistry data, these are fluorescence spectra, where we have wavelength on the x-axis and we have fluorescence, normalized fluorescence on the y-axis. And the different colors represent plastics that have different degrees of UV light degradation. So if it's purple, it's not degraded at all. It's not been weathered. But if it's red, moving towards red, it's much more weathered. And so we can see there's a shift in what the data tells us, and we actually can perceive a different change in color with a stain. And this was kind of our first step into building and growing our analytical capability to be able to look at the degradation of plastics and that we intend to apply to a microplastic detection tool as well. So kind of coming back to that idea of microplastics. And so I think one of the things that's kind of come up multiple times within my talk is that we really need and use a lot of instrumentation as part of this plastics research. And the awesome thing, so I put some pictures up here for folks that maybe haven't necessarily toured the Heikela Chemistry and Advanced Materials Science building. This building opened about 6 or 7 years ago, and as part of a new building being built, we had the awesome fortune to be able to equip it with sort of cutting edge technology that allows our students, both in instructional labs and in research labs, to have access to instrumentation that allows us to be kind of one of these leaders in plastics research. And that includes a variety. This is that microscope that I talked about, that fancy FTIR microscope that I talked about. We use the different chromatographs for different analysis of different additives that might be leaching for our plastics. We do some UV vis here. One thing I would say is that, folks that work in industry or work with chemical instrumentation, 6 or 7 years, we're starting to kind of come up to needing some maintenance or needing to think about replacing some of this stuff. So we are looking in our department to continue to be able to offer this access to this scope of instrumentation that really sets our students apart. Is that we have the UMD Eleanor Collins Chemistry and Biochemistry Instrumentation Funds that's been recently endowed and set up that is going to allow us to continue to maintain and provide the world-class research facilities that is kind of becoming the expectation for what students from UMD and from our department get to experience. So with that, I hope that I kind of spurred some interest in this area or let you know about some things that maybe you were interested in. I encourage you can keep up with my group at the QR code here to learn a little bit more, keep up with what our research group is doing, or if you, again, I put that QR code for the citizen science effort up there as well for folks that are interested. So with that, thank you so much for your attention.
Awesome. Well, thank you, Melissa, for sharing about this important research. We are now going to open it up for live Q&A. And so as a reminder, you can submit this at the bottom of your screen in the Q&A box, and we will try to answer as many questions as we can. So there were also some questions submitted in advance through our registration form. So we will try to answer those right now. So you talked, Melissa, a little bit about the emerging plastics. There was a question that came in wondering about the degradation process of these, and wondering how they are or how they can be biodegradable.
Yeah. So most of all of the biodegradable plastics or compostable plastics that are out there now are generally a polyester of some variety. And biology has been really good at evolving to be able to break ester bonds. And so some are better at breaking apart than others. So like lots of folks have maybe heard about a spinoff from Cargill called NatureWorks. They are the ones that really, I think, kind of commercialize polylactic acid, which is a product, lactic acid, they come from corn. And they were able to produce this compostable plastic. The issue with why these plastics are not maybe more widely employed yet is some of them really need composting conditions to degrade, and compost or industrial composting conditions, which is like elevated temperatures, really high microbial activity, so they can really get in and break those bonds. The other reason why these things have not necessarily been more widely deployed is the physical and mechanical properties of these biodegradable or emerging plastics maybe don't meet some of the expectations for what we use plastics for now. So, as an example, sometimes they're a bit more brittle than we want, or they maybe can't be foamed in a way. So, we use a lot of foam for like our couches and our mattresses and things like that. Um, that gets to be really, really tricky to then use these plastics. But the breakdown of these kind of biodegradable plastics is they need sort of high microbial um, activity, high enzyme counts. And so if you have if you have something that, if you have that compostable cup and you don't kind of put it in a compost condition, it's not going to break down very quickly.
Perfect. There are also several questions curious about health implications from microplastics. So you talked about physical risk, chemical risk, biological risk. Can you talk a little bit about kind of risk to our health?
Yeah, thanks. So I will say this is such an emerging area of research that in terms of the microplastics particles themselves and the impact on our health, I think we're only starting to learn where they're going in our body and how long they're there. I think if we eat larger plastics, in the mid micro size and whatnot, and we like accidentally ingest it, our body is pretty good at expelling those through our digestive system. The concern there is if there are kind of toxic pollutants absorbed to the surface or additives that leach out in this process, that would be a problem. The place that we're learning more about now is the presence of nanoplastics, which are small enough that it can kind of cross barriers in a different way, biological barriers in a different way, and translocate into our body in different areas. And so, folks, I'm sure have seen the popular the news, there's been evidence that they're finding nanoplastics in all sorts of different tissues and breast milk and placentas. And so um, we don't quite know what that means in terms of how our body is responding in a toxicity response to those specific particles. So I think the fact that they're there is important to know, and now we need to figure out at relevant concentrations what that means. Some studies have come out, they've used really high concentrations of micro and nanoplastics that they've exposed their organisms to. So it's hard to know, like, will that still be the case at kind of relevant physiological conditions?
There's also a question kind of related to this, curious about what manufacturers and politicians are doing, if they're doing anything to address microplastics in our daily lives.
So, I mean, manufacturers, the thing that there are rules through OSHA about exposure to that worker exposure to different things is something that is monitored as they're working on things. I think that is and particles and particle exposure, like inhalation to particles, is part of the OSHA kind of guidance. But in terms of policy, so um, there has been local and state policies around plastics and plastics use. There's many municipalities that have chosen to do like single-use bag bans, or where or you pay an extra 5 or 10 cents to get a plastic bag. There is evidence that having the 5 cent charge on a plastic bag actually reduces the amount of plastic bag waste that people see in the environment. So there's evidence that that is a potential impactful local level response. On the state, there's been efforts to remove microplastics from personal care products. And actually, I think it might be on a national level, even the microplastic bead ban. So you no longer can get an exfoliant in personal care products that would be made out of plastics. On the international level, there's been a lot of conversation about like the plastics treaty. This is like, can we, similar to sort of the climate change effort of like, this is kind of a global problem. How do we think about plastics and regulatory action on plastics on a sort of an international level? I will say there is not necessarily, like, much like the climate change like efforts. It's not like there's recommendations out there, but I think we're newer on our understanding of the impact of plastic, so they haven't gotten super specific. The last thing I'll say about regulation is that we are seeing that monitoring of microplastics in for as a water quality parameter is probably something that different like municipalities or lake associations or county boards are going to be taking on as an issue. And I would, this is me predicting. You could call my bluff, but I think in like 10 years, there's going to be an expectation that you're testing your waters for microplastics. So.
And that actually hits answers this question here. Just as consumers, how should we be aware of either our contributions or impact? And is it a bigger issue than just individual?
Yeah, so certainly the best like a lot of environmental contaminants out there, the best way as individuals we make an impact is just using less plastic, trying to tr make your choices if you do need new food storage containers, perhaps you go with the glass options, and I think that this has been something people have moved towards actually in the last 10, 15 years. Metal water bottles became a thing after we realized BPA was problematic, leaching from Nalgene bottles and the previous formulation of Nalgene bottles. And I think that that is going to be true too. Like you can make choices about what you use and the materials that they are and do your best. Now, certainly, I don't think plastics are terrible. I do like to eat strawberries in February, and those come in a plastic container, right? And so there are ways in which we don't avoid using plastics. And I think from a like a an effort kind of more on a community scale, um, the best way to prevent microplastics from happening is keeping the plastics out of the environment. And so community cleanups are really important in this. Things of that nature. And it's certainly advocating for good environmental policies on a state and federal level as well.
Perfect. Thank you. So there's a couple of questions that came in live, kind of more around the science and the chemistry and biochemistry of it. So we'll try to tackle a few of these. One question is asking, do plants uptake micro and nanoplastic particles?
Yeah, great question. They do. They do. And we're learning more about kind of what types of particles or what coatings happen on like what kind of things attach to the surface of the particles that may mask it more or less for the particle to move into the plant. Um, a lot of the studies I've seen have been primarily on like rice, and I'm trying to think of like food products that might do that. I don't necessarily think I've like seen something looking at apples or even corn or something like that. But we do know it's moving up. I'm not quite sure that we've seen evidence that it moves into the food yet of something that we would necessarily eat. But it could be, I mean, thinking about phytoremediation, maybe in the future we could think about plants that are really good about taking up particles. It might be a way to kind of clean up soils.
Well, there's a question that came in asking, what do we know about microbes that eat plastics? And do these offer promise to eliminate plastics in the environment?
Yeah, I think, well, maybe my Google news feed is more unique than other people's, but I feel like every other week I'm seeing like an article being a press release come out about a new strain of bacteria, a new species of waxworm that's able to digest and break down these materials. And I think certainly, I actually think that's a really promising potential way to get rid of plastic waste. I don't think we're at scale yet of scaling up, but I think that there's a lot. We have a lot greater tools for doing some genetic engineering, some of the CRISPR-Cas technology and things like that than we did even, 15 years ago. That this is going to be an exciting time to be able to think about maybe instead of, we have plastic codes 1 through 7, and many municipalities only accept like 1, 2, and 5 or something like that. And so maybe 3, 4, and 7 go to the bioreactor, and they've just enriched it with microbes that are able to chew away at that and get rid of it. So I could envision a world where that's going to be possible. And I think it's a really interesting area to pursue. Some of the work in my lab is actually trying to understand how the differences between materials properties and which enzymes are going to be more or less better at degrading these plastics based off of their materials properties. So I get really excited about that. Let's talk more.
Another question, wondering if there's an issue with bioplastics being placed in landfills due to the anaerobic nature of the landfill. Would this create methane?
I'm not sure. I mean, I think it would create methane. I mean, I think that you're probably working with a little bit easier to break down carbon sources that are creating methane at higher amounts, like the food waste that gets into the landfills. I don't the issue is there is the likely not going to be as active microbes that are able to hydrolyze those ester bonds to break it down. Um, so yeah, they don't typically break down in these landfill conditions. So that's why it's really important to know if you have a compostable material that you try to get into the compost area. Um, so,
Well, and there was a question, someone asked about should they be testing their soil for microplastics?
so testing your soil, geez, I mean, probably there are some microplastics there. To be honest, any place people have looked for microplastics, they've found it in different compartments. Um, I think that we're not quite sure if like what the impact is on sort of the growing plants necessarily at relevant concentrations. I think we're still trying to understand that. But there's certainly lots of questions about the how much microplastics are being put onto like agricultural fields as the and even coming from things like sludge, which is from the wastewater treatment plant. These are areas that are a big area of research. So I think I mean, I feel a little bad. I'm like, ask me in 5 years, ask me in 10 years. I think what you're seeing is that we're at the cusp of like learning a lot of really cool things and hopefully things that we're able to then address in the way that we have our farming practices or our gardening practices or things like that.
Well, and I think you mentioned that in a few years we all might be testing our water for plastics. Do you know if there's a way for homeowners to remove plastics from their water if they are present?
Yeah, great question. So, like there's actually some evidence that some of the water filtration systems should work okay that have like activated carbon and things like that. They're pretty good at removing the microplastics from it. It's kind of a little funny that they come in a plastic jug though, the Brita containers and stuff. Um, Drinking waters tend to be pretty good though for microplastics concentrations. Most of the current drinking water treatment tools remove most of the microplastics. And so you are going to be having to like drink a lot of water to get a substantial amount that you would be drinking. Assuming you're not using a plastic cup and assuming you're not, yeah, that you're not, your pipes perhaps are not all necessarily plastic, though I know we use a lot of PEX in our houses, and so that might change. But municipalities really good at removing. I don't think the danger is there in drinking water as high. The question is still out on well water. We don't have a lot of information about well water, and every well is different, we know. So we're working on, my colleague, Liz Miner, just, one of like probably the university's experts on microplastics analysis in natural systems. She's been working on some groundwater source analysis for microplastics. So
Great, thank you. And I think we will end on this last question. What is one of the most exciting things that you are looking forward to within your research?
oh, I mean, I guess I don't know if it became obvious through answering these questions, but talking about my research is like one of my favorite things and part of my day. I actually think that I get the most excited because I get to work with students. And so I turns out I can get pretty excited about any research idea that we come up and that we have engagement around. The things that we're really excited to look at right now actually fall very much in the fundamental chemistry, biochemistry world. Like, how can we think about the way molecules are changing on the surface of these plastics? And what does that mean in terms of how an enzyme is going to be able to attach and attack that bond and things like that? So those are the things I'm working on kind of most imminently with my students, but certainly just working with students is kind of like my favorite part of my job. So
I think it shows. We all feel it.
yeah.
Well, thank you so much, Melissa, for taking the time to visit with us today and share your expertise on this topic. And for those of you that are not UMD alumni, we host these sessions monthly. If you'd like to be added to our webinar series, feel free to send us an email. As Melissa mentioned, there was a QR code on one of her slides. I will be sending you all a follow-up email with a link to the Citizen Science Project, as well as a link to her research web page as well, if you'd like to dive in a little deeper. And Melissa also mentioned that she's open to continuing the conversation. So if anyone would feel like they want to reach out directly to her, she offered that up as well. So I think with that, again, thank you, Melissa, and thank you for all joining us today. And we hope that you'll join us again for future Bulldogs Behind the Scenes. Thank you.